Last week we talked about protein synthesis, protein modifications, and we also saw that proteins cannot be produced, modified, or carried without the other help of the other proteins. So there must be proteins for protein production, protein modification, and protein transportation. And we will talk about today the last step of the protein production, actually the modification. It is protein trafficking, which takes part in the Golgi apparatus. After the modifications which are done in the endoplasmic reticulum, protein is sent to Golgi apparatus. These proteins may start to work in Golgi apparatus, or they be modified and a ticket is given to them and sent to outside of the cell or lysosome. And there is something very interesting about Golgi apparatus. It doesn't have a stable body. We say something Golgi apparatus, but it is actually not Golgi apparatus, but it is Golgi apparatus. We will see how it is happening. The proteins which are transferred from endoplasmic reticulum to Golgi apparatus is transferred in vesicles. These vesicles are little containers. Actually, we are talking about containers, but they are little bubbles. You talked about the cell membrane, the bilipid layer last week. It is bilipid layer again, these bubbles, these containers. It is fluid, like olive oil, a bubble inside a bubble, and it is conscious bubble, actually intelligent bubble. It doesn't burst or it doesn't break apart, but it is fluid. It carries its load carefully. And this is type of endocytosis. Proteins are transferred safely in these bubbles because they shouldn't touch with water. The inner part of the cell is filled with water, not simply water, but the main ingredient is water.
Yes, and a Golgi apparatus is considered as a post office in the cell, because the Golgi apparatus receives proteins from one place, yes, from one address, and targets them to for delivery to another address.
Yes, and let's talk about what's happening in Golgi apparatus. First, vesicles comes to cisternae. Cisternae is the nearest part of the Golgi to endoplasmic reticulum. Vesicles fuses with cisternae and release their packages. These vesicles form the new cisternae. At this point, we have to talk about a cis maturation model about Golgi apparatus. A new cis Golgi is formed by the fusion of the vesicles that come from the endoplasmic reticulum. Cis Golgi maturates to become medial Golgi. And maturation process includes retrograde vesicular transport and resident Golgi proteins. As the proteins move, they are modified by the Golgi enzymes at specific locations in the Golgi.
Each cisterna comprises a flat membrane, enclosed discs that include special Golgi enzymes, which modify or help to modify the cargo proteins that travel through it.
Yes, yes, and the enzymes add carbonitrate and phosphate to the proteins in modification process.
So how does the proteins move through Golgi apparatus? The movement is in waves actually. First, the cis cisterna becomes a part of the medial Golgi. Behind it, a new cis cisterna is formed by the fusion of the vesicles from the endoplasmic reticulum. Meanwhile, one of the medial cisternae migrates and becomes the new transcisternae. This is cis maturation model. Proteins are sorted within the trans-Golgi network according to their added signals. Proteins with the same target sequence are destined for the delivery for the same location. Then the vesicles start to come out from the trans-Golgi network. These vesicles then migrate to target location. Then their proteins may go to outside of the cell, as I said, or lysosome. Proteins prepared for secretion after the modification in the Golgi are transferred from the trans-Golgi to the plasma membrane. When the membranes fuse, these proteins are released into extracellular space. Other proteins, including lysosomal enzymes, are sorted to lysosomes from the trans-Golgi, and lysosome is the digestive organelle of the cell.
And I want to give an extra information about Golgi apparatus. In the animal cell, the Golgi apparatus will break up during disappear during the onset of mitosis. or cellular division. And during the telophase of mitosis, the Golgi apparatus reappear again. And as of December 209, it is uncertain how this occurs.
Yes.
And but it is different in plants or yeast cells. Golgi remains inactive in these. The reason for this is still unknown for the scientists.
Yes. This is amazing. It disappears and come back immediately.
It's very amazing.
Yes, amazing. This is, they say this evolution, this is beyond evolution. Evolution is impossible. We say here that it doesn't, Golgi apparatus doesn't have stable body. It has proteins. Proteins are modifying the coming proteins and giving tickets to them. Giving ticket is something not very easy because it is not just giving a ticket. It makes chemical changes on them.
Yes.
It's not something
you should
very
know
easy.
the coordination. You should know where it is going, who it will be. Going for and what it will do. You should know its purpose, why it is produced, what this package is containing, and you should give the ticket according to its the function.
Yes,
you should know it.
And this maturation model, as we talked about, it is not growing uncontrolled way. When the new vesicles come, the one part of the Golgi apparatus go away takes the proteins to their destinations, and these are done. These vesicles doesn't come to endoplasmic reticulum. It doesn't come to Golgi apparatus or go away from the Golgi apparatus by it themselves. The proteins are carrying them.
Yes.
And these proteins have special rods formed of proteins, and these proteins are carrying them by walking on that road and carrying them on their heads. Actually, they are not walking; they are running because in the cell, 2,000 proteins are produced in the cell in a second. So they have to run. Everything has to be very fast, and we cannot see them with our eyes. It is impossible because it is so fast.
Yes, they have a different time for us.
Yes, mashallah. And this is not evolution. There is supreme creation. There is a perfect system and order here. Yes. And this is all creation of Allah, and all the things that I talked about here, the bilipid, the proteins, and the other parts are just the molecules, but they are acting consciously. This is the conscience of Allah.